So first of all, huge thank you to Sheila, Carolyn, and the fantastic folks in the Ecology, Evolution, and Conservation Group at York University. I'm so honored to be presenting at BeeCon. And so what I thought I would do today is, instead of focusing on one specific project, in honor of all the wonderful grad students that are presenting in this conference, I thought I would talk a little bit about what I started working on as a graduate student and how those projects have kind of evolved over time. So with that, the title of my talk is Plant-Pollinator Interactions and Ecosystem Services in the Face of Global Change. And I really want to focus on this relationship between biodiversity and ecosystem function. And so I'll start my talk there. Thinking about biodiversity and ecosystem function, this inquiry, this area of research has really just exploded in the last 20 years, starting with kind of classic studies of biodiversity and ecosystem function when it comes to responses such as productivity or stability, and experimental work, including work by Dave Tillman and others. And really, that much of that work served as a foundation for us to think about species, not only in terms of their number, but also their functional groups or roles. And then a lot of that work has really been, I think, fundamental in laying the groundwork for connecting ecosystem function with ecosystem service. And some tremendous work by Gresham Daly and the Millennium Ecosystem Assessment has really shown us the diversity of services or benefits human experience from these valuable ecosystem functions. And now more recently, there's been a tremendous amount of work by Velland and Tilianakis and many others that have really shown us that this relationship between biodiversity and ecosystem function, also known as the BEF relationship, is very scale and context dependent. And it really depends on how we define biodiversity. And so in my talk today, I want to take a closer look at that relationship between biodiversity and ecosystem function. And obviously, this is BeeCon, so it's going to be much more focused on plants and pollinators, and specifically plant and pollinator biodiversity influencing ecosystem function. So in this image, we have a Meliponine bee, a native tree, coffee bush, as well as a landscape that's experiencing a lot of soil erosion. And we can think about these tropical plants and pollinators providing pollination function as well as erosion control. But I really want to argue in my talk today that we need to move beyond the species concept in biodiversity and really look at some of these intra-specific factors, such as behavior, genetic and genomic features, morphological traits, and their variation within species. And so I'm going to walk us through this figure as kind of an orientation to the rest of the talk. And I really hope I'm able to make the argument today that it's really important for us to think about how pollinator foraging dynamics also influence ecosystem function, how plant and pollinator landscape genetics and genomics influence ecosystem function. And finally, if we really want to understand plant and pollinator interactions and their impact on ecosystem function in the context of global change, we need to be doing this work across heterogeneous environments, really exploring interactions between local and landscape management, as well as potential impacts of climate change. So I started with this tropical ecology system to outlay kind of my framework for this talk. And the reason I started there is because that is how I was introduced to this world of research in my graduate program. So when I started in grad school, I really was interested in this plant and pollinator interaction and how it impacted ecosystem function within coffee agroecosystems. And I wanted to build upon that and really explore the influence of pollinator foraging dynamics, plant and pollinator landscape genomics, as well as these global change forces. So starting in that coffee agroecosystem study system, I'm going to just orient you to kind of the classic way we think about coffee management. And so on the left side here, you can see at the top, we have our more rustic or shaded traditional management, where there's a large diversity of overstory trees, subcanopy trees, and then you have coffee bushes in that bottom subcanopy, below subcanopy layer. And then as you move downward on this visualization, what you can see is we move towards the more modern and unshaded management types for coffee, where large overstory trees or shade trees have been removed, the coffee bushes have been increased in density, and in general, this is a less traditional approach that has some pretty major implications for biodiversity. So when I started working in this system, about 43% of total world coffee management in the world was in this diverse shade style, what you see at the top of this chart. But by the time I had kind of finished my PhD, that number was dropping precipitously. And we can see it happening around us in southern Mexico and elsewhere. And now currently only about 24% of coffee cultivation is managed in this rustic traditional style, despite the fact that these biodiverse coffee habitats provide some incredible habitat for migratory birds, more than 150 species just in some of the cocoa farms I worked in. And so my real question was, how does this influence coffee pollinators? And I started my work in that question, exploring that question, and found that indeed shade promotes pollinator foraging activity for native bees, but alters or has a completely different influence on how exotic honeybees actually perceive resources in the landscape. And further, shade tree diversification can actually drive native bee diversity in these shade coffee systems in southern Mexico. But as I was working in this system, I became really fascinated with the shade trees themselves, and in particular, one species, Miconia affinis, which is one of the most specious genera in Central and South America. And it has a very specialized pollination system like many tropical trees do, which is that it's buzz pollinated. So the anthers are shaped like tubes with an apical pore on top. And in order to get the pollen out, bees, and specifically native bees, have to grab on and vibrate their body. So the non-native honeybees are not capable of this sonication. And so I was really interested in how landscape management influenced the specialized pollination system, using this species as a sort of bioindicator of how native bees are moving pollen across these landscapes. So how do you actually measure pollen movement? Well, it turns out it's not that different from how you measure siring across spatial scales in human populations, which is you use genetic or genomic tools to conduct a parentage analysis. You pull fruits from a mother tree, you know the mother's genotypes, and then if you've assessed all of the population or as much of the population as possible, you can deduce who the father is and actually map out pollen movement. And so when we did this in southern Mexico in the shade and coffee kind of mosaic habitat, what we found here is we look at pollen dispersal events on the y-axis, so number of events on the y-axis, and the actual dispersal distance here on the x-axis. What we can see is that mother trees in the forest and mother trees in the coffee have very different pollen dispersal kernels, where the mean dispersal is greater for mother trees in coffee compared to forest. And that's indicated in blue here. And both of these distances are far beyond nearest neighbor distances. So pollinators are moving much further than they need to, much further than the nearest tree. But I think what's really striking is the fact that there's just a lot of cross-habitat gene flow. So between these coffee farms and forest fragments, there's a lot of pollinators moving between and basically spreading pollen across habitat types. So the next question after I finished my PhD work was really thinking about how other aspects of, again, biodiversity beyond species identity could influence this plant reproduction or ecosystem function. And specifically, what could the impacts of local kinship and pollinator community composition be? And so I moved this system from southern Mexico to Panama, and largely because moving to Panama and extending the system there allowed us to really look at a lot more landscape heterogeneity and a lot of variation in local kinship, and allowed us to really incorporate these into our concepts of pollination ecosystem function. So I couldn't have done this without the tremendous work of my graduate student, Megan O'Connell, and postdoc Antonio Costilla. So we set up a system in Panama with 11 study regions, each with finely mapped adult populations, adult trees, where we could look at density dependence as well as local kinship. And what I mean by local kinship is fine scale spatial genetic structure. So in this example here, you can imagine each of these trees is representing a different genotype or color. In this case, and this would be what I would say was a low kinship neighborhood versus a high kinship neighborhood where all of the trees in the cluster are highly closely related to one another. So we explored the role of this local kinship. And in a subset of these landscapes, we also conducted these pollinator exclusion experiments where we focused on the impact of a single pollinator in terms of seed set as well as pollen dispersal. And then in another subset of landscapes, we actually were able to compare the same trees in an El Nino year versus a typical year. So the exact same maternal trees, looking at the pollen dispersal and seed set patterns across those 2 years. And the first thing I want to show you is that indeed, as we might expect, pollinator body size does predict seed set. So the smaller pollinator species like the Tetragonisca angustula have lower seed set or set lower seed for Miconia than Melipona panamica. Now the question was, is this true also for pollen dispersal? And what we found is in fact, pollinator body size does not predict pollen dispersal. So our largest bee, Melipona panamica, which is about the size of a school eraser, had some incredibly long pollen dispersal distances, almost up to 3,000 meters. But even our smaller bodied bees like Trigona fulviventris, Trigona fusipenes, and Tetragonisca angustula also had incredibly long pollen dispersal distances. And so thinking about how large these are, this is about the size of a ladybug, a grain of rice, and a lentil. So these are tiny, tiny bees moving pollen tremendous distances. So pollinator body size affects seed set, but it does not affect pollen dispersal. What does affect seed set in addition to pollinator body size is this panel on the bottom here, where seed set is clearly a function of local kinship. And that's that factor I mentioned before, where if you have a low kinship neighborhood, you're going to have higher seed set because the pollen that's being deposited on you is from trees with distinct genotypes. Whereas if you have a high kinship neighborhood, you've got a lot of biparental inbreeding going on. So what was astonishing is the fact that this local kinship had a really dramatic negative effect on seed set beyond pollinator body size, right? Now, our next question was, do we see similar effects of local kinship across different climate change regimes, specifically in an El Nino compared to a typical year? And what we see is that same pattern of negative effects of local kinship on seed set, but that pattern is much stronger in El Nino, significantly stronger. And so what that means is in extreme climate conditions, the negative effect of being around closely related trees or closely related individuals is even stronger, right? And when we took a look at why this might be happening and looked at the pollen loads of the pollinators that are mediating these reproductive events, what we see is that in our ENSO, or El Nino year, pollen fidelity is much lower and much more variable compared to our non-ENSO, our typical year. And that if we broke this down further and actually looked at the plant-pollinator network interactions, indeed we find that same pattern is true for network specialization. So the more, so in an El Nino year, pollinators were far less specialized than they were in a typical year. All right, so I've given you a little overview of this tropical study system, and some key take-home messages or findings from this work are that bees engage in frequent cross-habitat foraging. Body size predicts seed set, but not pollen dispersal distance. Local kinship, or in other words, low genetic diversity at that local scale, fundamentally drives plant reproductive success. And we think this has to do with the fact that so many pollinators, including these native meliponine bees, are local trapline foragers. So they forage very locally, and thus local kinship is playing this incredibly important role in plant reproduction. And finally, high landscape genetic diversity, or that higher level of genetic diversity at that local scale, can buffer the negative effect of extreme climate on plant reproduction. So local genetic diversity becomes increasingly important in the context of climate change. All right, so now I'm going to shift over to temperate systems and orient you with the same visual here. I'm we're still going to talk about the implications of biodiversity for ecosystem function, but I really want to focus on pollinator foraging dynamics and how those have been impacted and continue to be impacted by climate change, as well as local and landscape management. So what you'll see here is we're working in temperate grasslands as well as cotton agroecosystems. And I'll start with talking about some of our temperate work in California, which grew out of my postdoc work. And this work was conducted just around the UC Davis area, where we focused on the species Bombus bosnicensis. And we basically conducted a landscape foraging analysis where we worked in 8 different study regions, each ranging from about 5 to 99% natural covered. And within each of these, we have 5 sampling sites where we collected bosnicensis. And because we collected them along a transect, what we could do is when we collected about 20 workers per site and also conducted vegetation surveys and GIS mapping, we could use molecular tools to actually examine colony identity and foraging patterns. And the way we can do this is, of course, using genotypes to identify colony identity. But then once we have colony identity, mapping out approximate foraging clouds by saying, well, if we have a forager that's 50 meters away from another colony mate, then at the minimum, you're foraging 25 meters from their colony origin. And we can continue to build on that as we recapture more colony mates. And we've done a lot of simulation work showing that this, in fact, is a good proxy for colony foraging distance. All right. So just with that nesting density information, what we were able to see in the system in Northern California was that nesting densities are largely driven by natural habitat cover, as well as impermeable surfaces, but that these act at very different spatial scales. So looking at both the 250-meter fine local spatial scale, as well as that 2-kilometer large spatial scale, what we see is at the regional scale, there's a positive effect of natural habitat. So on the y-axis here, we have percent of nesting variation explained. And on the x-axis here, different regional land cover types, including crop, grass, orchard, bare ground, impervious, oak, and riparian, oak woodlands and riparian forest, where blue bars indicate positive effects and black bars indicate negative effects. So at a regional scale, positive, significant effect of oak woodland habitat on nesting density. But at that finer 250-meter scale, what is a very important and significant predictor of nesting densities is actually impervious cover. So the amount of impermeable surfaces has this negative effect on nesting densities. Using those foraging clouds, foraging mapping, what we're able to see in this landscape is that foraging ranges are very, very dynamic. And they're not influenced so much by habitat land cover, but by floral resources in that landscape. So specifically on the y-axis here, we have colony level foraging distances. On the x-axis, we have landscape floral cover, coefficient of variation, so how much variation there is in floral cover. And on that diagonal z-axis, we have local floral richness. And what you can see is that colony level foraging distances increase with floral species richness, but decrease with landscape level floral heterogeneity. So in other words, these bees, these bumblebees, Bombus bosnicensis, are foraging further for high diversity patches, but they're also doing this in more consistent and homogeneous landscapes. Why would they forage further in more consistent and homogeneous landscapes? It actually makes sense when you think about the fact that many bees and many small-scale foraging studies have shown that bumblebees are risk averse. So when they encounter heterogeneity, they're less likely to take longer distance foraging risks. So our next question was, well, is this true across many seasons? So that first study, we just did it during the peak bloom. So our next study, which was led by my student Nick Pope, we looked at both the peak bloom as well as the low bloom period and found that that high selectivity for patch quality became even stronger in the late season. And what we think is happening, the reason why we're seeing longer foraging distances and greater patch selectivity in the late season is because this is when the colonies are starting to senesce, but the individual impact of each of those foraging events is really critical for new queen and male survival. And so we think that what we've been able to reveal here is a really important period of foraging for colonies in this later season. Okay, so using those same bees, we can also look at the genetic structure of these bees across much larger spatial scales. We expanded our study to include almost all of the West Coast. And what we can see with our focal species, our original focal species, Thalamus bosnicense, is that indeed the genetic structure of this bee shows that, indicates that this genetic structuring is representative of potential resistance to urban or crop habitat. So basically what we did is we created a circuitscape-based resistance map where we can calibrate land use types to represent potential resistance to dispersal processes. And though genetic structure is representative much more than just dispersal, also genetic drift and other evolutionary demographic processes, this can serve as an interesting way to look at potential signatures of dispersal limitation. And what we see is that in addition to classic isolation by distance patterns, we see an additional significant effect of this resistance landscape. So on the x-axis, we have resistance distance, where greater resistance to urban and crop cover is indicated by moving further right on that axis. And what we see, in fact, is that these populations are structured by urban and crop land use, beyond just isolation by distance. What was fascinating, though, is that we started with this species and spent a lot of time working on it. When we moved to other species, for example, Tetragonics angustula and many other stingless bees in Central and South America, what we found is that this pattern did not always hold. So sometimes there were no effects of land use or even facilitative effects. So in a study conducted with my colleague Rodolfo Jaffe and others, we looked at 17 different stingless bee species and found only isolation by distance patterns in some species. And that isolation by distance pattern really depended on whether the species had been domesticated or not. So if a stingless bee had been domesticated for honey extraction, then that isolation by distance sign would also disappear and that line would become flat. In other work we conducted in Texas, specifically my postdoc Kim Villar did this excellent work looking at Xylocopa virginica across these urbanizing landscapes and found that, in fact, we see this opposite pattern where urban habitat facilitates gene flow for Xylocopa. And what we think is happening here is that because Xylocopa virginica is known to nest in human modified landscapes, and wood piles and construction sites, what we think is going on is and based on the genetic structure and patterns across highways and intensely urbanized areas, is that many of these urban habitats are serving as just corridors for gene flow as these wooden products are moving between cities. Also, with this genetic data for Xylocopa virginica, we could look at finer scale kinship in the same way that I described kinship earlier, and examine this relatedness that's now on the y-axis here, so kinship or relatedness index, and we have geographic distance on the x-axis. And what we can see is that just at less than half a kilometer, so that within 500 meters, individuals of Xylocopa virginica that are not siblings, these are fully non-SIV individuals, are yet still highly related at this fine spatial scale. So we've got basically relatives, cousins that are all staying close to their natal nest sites, and more than you would expect at random at this fine spatial scale. And this lines up with what we know about carpenter bees and their tendency to go back and refurbish old nest sites, but to actually see it in the fine scale kinship data was really exciting. All right, so one of the realizations as we were conducting some of this population genetic and genomic work is that there are a lot of biases in the data. And so with my colleagues Margarita Lopez-Uribe and Antonella Soro, we decided to conduct a little meta-analysis looking at population genetic and genomic work, and potential roles of things like body size and sociality. And so what we did find across over 50 studies and more than 40 species, we found that indeed body size and sociality immediate low levels of genetic structure. And the larger body size you have, usually the less genetic structure you'll see. And that social bees tend to have lower genetic structure than solitary bees. But what we also recognize is that in these studies, there's a limited utilization of next generation sequencing tools, largely because of some of the challenges of using historic specimens, which have been widely used in microsatellite or other molecular tools. And so all of our research groups have been really exploring how we can take advantage of highly degraded insect DNA. And there's a lot of opportunity with tools like DBRAD for exploring larger scale population genetic and genomic structure, even for historic samples. So finally, what we recognized in this meta-analysis was that the past work has really focused on corbiculate bees and has really tended to focus on social species. So we really need to be pushing the boundaries more on non-corbiculate solitary species. And because of that, we've started some recent projects looking at Melisodes across the US, as well as in South America, focused right now on Brazil, with our collaborators, Ken Young, Maria Zucchi, and students Sarah Kusser, Kim Balar, Caroline Miranda, and Nick Pope. And the reason we focus on these Melisodes species is really important for cotton pollination. And so our work in cotton pollination, just in Texas, has really revealed a lot of interesting patterns related to functional roles in the pollinator community. So although when we started this work, cotton farmers were not really convinced that pollinators were playing any role in their systems, what we've been able to show over many years is that not only do bees play a role, but even butterflies and flies are actually acting in complementary ways, foraging on different nodes of the cotton plants at different times of the year and day. And so there's incredible amounts of functional complementarity occurring due to this broad suite of cotton pollinators. And we see that when we look at pollen limitations. That's a figure here on the y-axis. We have basically limitation of how different a site is from its optimal level of pollen deposition and pollinator abundance, and on the x-axis. And so you see, as pollinator abundance increases, our overall levels of pollen limitation decrease, meaning that those fields with high pollinator abundance and pollinator richness are getting closest to optimum pollination service. And so although the pollinator community broadly is providing this, we also found that one species in particular in Texas was really critical, and that is Melisodes typanica, a longhorn bee. And so using a series of different hand pollination studies, what we could see is that when we compare our no pollination group and our out cross pollination with our self pollination group, seed cotton weight is dramatically higher. And so overall, what we see is about a 17% yield increase when we have out cross pollination. And just in our study system in South Texas, that's worth over $1 million. The value to Texas more broadly is something we're working on calculating and spatially mapping out at the current time. And we're also really interested in asking whether these pollinators, if managed properly, could be providing even greater yield across other regions of the world. And so far in our work in Brazil, we've noticed that indeed there's another longhorn bee in that system, native Melisodes nigroana, that seems to be playing a similar role. So we're really excited about further pursuing better understanding the impacts of these key pollinator species in these pond systems. Okay, so I've told you a little bit about ecosystem function in cotton systems, but now I really want to dive deep into this area for the last portion of my talk. I started this talk really thinking about the biodiversity ecosystem function relationship and how important it is to incorporate foraging dynamics, plant and pollinator landscape genetics, as well as global change forces. But another important feature of understanding ecosystem function is really not looking at any of these functions in isolation. And so the biodiversity and ecosystem services or biodiversity and ecosystem function relationship, as we explore it, it's really essential to move beyond single indices such as just pollination service and really understand potential trade-offs and underlying mechanisms for a variety of different functions and services. So for example, if we have this figure here where this is work from my graduate student Kim Villar's dissertation, where she looked at urban gardens and urban green spaces and found that bee species richness declined with increasing amounts of leaf litter or ground cover, such as mulch. And this is something that many other urban pollinator studies have found, that when you cover the ground and prevent ground nesting opportunities, indeed your bee species richness decreases. Now, our question, if we have decreasing bee species richness and pollination service with increased litter, but we know that this litter, this mulch, this ground cover can actually increase climate buffering or water conservation, how do we optimize multiple ecosystem services, multiple ecosystem service providers in the same landscape? And what are some of the underlying management mechanisms that we need to promote to do so? So this is work that incorporates pollination, pest control, carbon sequestration, water availability, climate regulation, and well-being. And it's work that I've been conducting in collaboration with Stacy Philpott, Brenda Lynn, and Heidi Lide in urban garden systems in Central California, as well as in designed urban green spaces here in Texas and Austin and Dallas with Amy Belair from the Nature Conservancy. And finally, in degraded and restored prairies in Texas and Oklahoma with Kristen Baum and Eleanor Lichtenberg. So for today's talk, since we don't have too much time left, I'm going to focus just on urban sustenance gardens and some of our findings there. And so again, looking at multiple indices, moving beyond a single index of ecosystem function or ecosystem service, what we see in these urban sustenance gardens, and this is work conducted across 28 gardens over multiple years, where we measured pest control, pollination, soil and climate and water ecosystem services, food production, as well as gardener well-being. What we found is that overall synergies tend to dominate across these different ecosystem services. So each of these categories includes many different services, which is what are numbered along the diagonal and along the top row here. And what you can see, this is a correlation table here, where blue represents positive relationships, red represents negative relationships, and I've only visualized the significant ones. But overall, about 70% of the significant correlations were in fact positive. So a lot of synergy across ecosystem services. And if we just focus on the food related ecosystem services, given classic sort of assumptions that biodiversity is at odds with food production, what we see is actually 80% of the correlations with food and other ecosystem services are positive. And this is also true when we include ecosystem service providers, so things like pollinators, insects, pest controlling insects, birds, and other ecosystem service providing insects. They all exhibit, for the vast majority, we see potential for synergy. And we see very few trade-offs with putative ecosystem service providers. So overall, a lot of synergies are dominating these landscapes. And although I'm only presenting the work from the urban garden system, this is really similar to what we're finding also in urban designed, non-sustenance, non-food related gardens as well. So potential for synergies across many of these key ecosystem services. Our next question in the system was really asking how local and landscape management can interact. So some classic work by Chanka and colleagues and has been really essential and helpful in understanding multi-scale impacts of habitat management on insects and on biodiversity or ecosystem service providing organisms. And this work has indicated that you can have very different patterns in response to local management depending on landscape composition. So if you look at the right side of this figure, if you have a locally simple habitat versus a locally complex habitat, within a simple landscape, they can be dramatically different in terms of how they support, on the y-axis here, a biodiversity or ecosystem function index. Whereas in a complex landscape, you might not see such a strong difference in the effect of that local management, since so many of those species are supported at that landscape scale. And so this has been part of kind of this early theory about local and landscape interactions, much of which has assumed that dispersal comes from the landscape into the local habitat patch, where you might be altering local management, for example, herbaceous plant richness, like in this diagram here. But what this theory doesn't take into account, and increasingly recent work has really tried to expand on this and kind of develop alternative explanations for patterns that we're seeing. For example, if foragers actually include a broader range of species, a broader range of ecosystem service providers, and if they behave in ways that actually follow forager dilution patterns. So if you disperse to the landscape from the local habitat patch and exhibit forager dilution, you might see something like these blue lines here, where in complex landscapes, the benefits of a complex, simple local landscape or a complex local landscape might be lower. You might have lower biodiversity ecosystem function if in fact these foragers are dispersing out into the landscape. So there's really a lot of variation that we want to try and capture. And so we went ahead and conducted this analysis across our urban garden system and found really striking evidence of both of these patterns happening. So both dilution and concentration effects in our foragers. So if we just look at the ecosystem service of pest control on the left-hand panel here, and ecosystem service providers of pest control, the natural enemies, as well as pollination on the right-hand panel, and pollinators on the right bottom panel, and how they respond to this local herbaceous plant richness, our local kind of complexity index on the y-axis, in these two different types of habitats, more simple habitats versus more complex landscapes. So simple and complex landscapes, what we can see is clear dilution and concentration effects. Right? So there's not a specific pattern. There's a lot of functional variation in how pollinators and natural enemies are inhabiting these landscapes, responding to local landscape resources, and affecting pest control and pollination. So now that I've told you a little bit about this temperate work, I want to just give you some take-home messages from this work and then take a further step back and talk about some broader, what I believe are next steps based on better understanding this biodiversity ecosystem function relationship. So just some key findings from this temperate work is that what we can see is that we have longer bee foraging distances in species rich, in terms of the plant community, species rich and evenly distributed floral resource landscapes, longer and more selective foraging in the late season. Urban and cultivated land differentially impacts bee genetic structure patterns, as does domestication, body size, and sociality. And this is what we've seen across these different meta-analyses, that there is a tremendous variation in how bees are responding to landscape composition, often based on body size and sociality, but not always. Synergies dominate in urban gardens and urban green spaces, where biodiversity and ecosystem services are not necessarily at odds with food production, despite long-held theories that they might be. And that many local and landscape interactions exist across this broad suite of ecosystem service providers. So this really highlights the importance of local complexity in these more urbanized contexts. So what are some key next steps given this broader array of interactions and how we can think more broadly about biodiversity? So some next steps in biodiversity and ecosystem function, especially given global change patterns. The first I feel honestly is that pollination function is very multifaceted. It's not just about seed set or pollen dispersal, it's about a broad suite of traits. And each of these can be mediated by different factors. What we've been able to show is that seed set and pollen dispersal can be mediated by both spatial and genetic structure in plant populations, as well as the pollinator community, their fidelity patterns, and global change patterns, so impacts of climate. So we really need to develop a broader understanding of functional diversity within each of these groups, so for both plants and pollinators. Pollinator foraging is complex and dynamic, so we really need to move away from measuring pollinator foraging at single time periods and really move towards quantifying the importance of resource availability and those resource distributions, as well as seasonality or the timing of a particular foraging event, to understand how these foraging patterns are shifting across the season and in response to local floral resources. Large-scale patterns of pollinator genetic structure indicate that body size and sociality can influence this structuring, and also that human domestication over thousands of years can influence this genetic structuring and eliminate patterns of isolation by distance that we would otherwise see. But we really need to move beyond the dominant taxa, move beyond corbiculate bees, and really start looking more closely at solitary bees. And finally, what we see is that many traditional landscape-mediated synergies exist in urban green spaces. So there's a lot of opportunity for optimizing multiple ecosystem services, but it's important for us to understand these interactions, and where we do see trade-offs, develop guidelines for avoiding them. So particularly in the case of food and biodiversity, evaluate how to best manage these local landscapes to optimize both. So with that, I want to acknowledge the current ancestral land stewardship in many of our research systems and at UT. So we acknowledge our presence on the traditional, ancestral, and unceded seized territory of the following groups. So in Texas, the Wichita, Tawakoni, Caddo, Cualtican, Sana, Cumanos, Kickapoo, Lipanapachi, Tonkawa, Comanche, and Osage groups. In California, the Tamien, Amamutsan, Awaswas, Ayuani, Patuin, Wapo, as well as the Kul and Kata in New Mexico. And so we strongly support the increased recruitment and support of Native American and Indigenous students, as well as first-generation students and many other underserved groups. Some of the work that we're trying to do to increase engagement beyond our traditional stakeholders, we recently were funded by U.S. Fish and Wildlife to develop a number of pollinator restoration activities. We started this work on public and private land, and are now hoping to expand this into some tribal lands. And this is being done via prescribed burn and wildflower seeding, but also in combination with some workshops we're providing in terms of native bee identification and some sustainable ag workshops we're providing for the cotton growing community, in collaboration with Xerces, Texas Parks and Wildlife, the Lady Bird Johnson Wildflower, and the Lady Bird Johnson Wildflower Center. This is also related to work that we started a couple of years ago, providing tax exemption and supporting tax exemption programs for folks, land stewards who are interested in managing for pollinators. So with that, I want to acknowledge my collaborators, my colleagues in the Philpott Lab, the Lynn Lab, Lide Lab, Lichtenberg, BOM and Muth Labs, the Brosi and Panosia Labs, as well as Jack Neff and Alonso Santos. Funding from the National Science Foundation, Army Research Office, USDA, U.S. Fish and Wildlife, National Geographic, and Texas Parks and Wildlife, in addition to the Winkler Foundation. And it's been an honor to speak with you all today, and I would be happy to take any questions you might have.